Hello my friends, this is lecture number 5, and in this lecture we will talk about the posterior pituitary hormones. Some of them you are already familiar with them. We'll go to some further details, and I hope it should be an easy lecture, and I'm sure that you will like it, hopefully. This slide remind you of the physiological anatomy of the pituitary gland. This is the posterior lobe right here. The posterior lobe, unlike the anterior lobe, does not synthesize the hormones. It just stores and releases the hormones, and its hormones are synthesized in the hypothalamus in two nuclei, the supraoptic and the paraventricular nucleus. These neurons send their axons down till they reach the posterior pituitary gland through this neuronal tract. We talked about this. I hope you can review it. This is the hypothalamus, and these are the two nuclei that synthesize and secretes the hormone down into the posterior pituitary gland. See, the hormone is synthesized here. When there is a proper signal, these neurons will synthesize these hormones, and the hormones will travel through the axons until they reach the posterior pituitary, where they will be stored. The two nuclei are paraventricular nucleus and the supraoptic nucleus. The supraoptic nucleus secrete both hormones, and the paraventricular nuclei also release two hormones. However, one of them, the supraoptic nucleus, mainly synthesize ADH or antidiuretic hormone, and the paraventricular nucleus mainly synthesize oxytocin. So there are two hormones, ADH and oxytocin. And remember, this tract is referred to as hypothalamic hypophyseal tract. Hypothalamic hypophyseal tract. And it goes through the infundibulum or pituitary stalk until they reach their final destination, which is the posterior pituitary gland. Now these are the two hormones again. Oxytocin is mainly synthesized in the paraventricular nucleus, and ADH or vasopressin is synthesized in the supraoptic nucleus. And you can see the major target organs for oxytocin is the uterus and the mammary glands, whereas the target organs of ADH, the kidney, blood vessels, arterioles, and it also there might be another site which is the sweaty glands, the seminiferous or sweaty glands. These are the two major targets of ADH though. Now let's it's summarized here. Posterior pituitary store and release antidiuretic hormone and oxytocin. You want to remember that the hormones are synthesized in hypothalamic nuclei. After they are synthesized, they are bagged in secretory granule with their respective neurophycins. They are pre-hormones then. Both are nanopeptides. Both of them are synthesized in the supraoptic and paraventricular nucleus of the hypothalamus. And as I said, ADH is primarily synthesized in the supraoptic nucleus, whereas oxytocin is primarily synthesized in the paraventricular nucleus. Now let's talk a little bit about the synthesis and the processing and the secretion of ADH and oxytocin. These are the hypothalamic nuclei. Let's say this is the supraoptic nucleus, which makes primarily ADH, and this is the paraventricular nucleus, which primarily make oxytocin. Like other hormones, they are initially synthesized in these neurons as a pre-prohormone. The pre-prohormone oxytocin is the pre-prohormone for oxytocin, and the pre-propricin is the precursor for the vasopressin or ADH. Once they are synthesized and bagged, and by the Golgi apparatus and secretory granules, they are cleaved further into smaller units, namely probrezophycin and pro-oxytocin, and they travel down via the hypothalamic hypophyseal tract, the axons, by axon transport, and they remain in the vesicles in the form of oxytocin and the neurophysin 1 and ADH and the neurophysin 2. And when we have an appropriate signal to the hypothalamic neurons, the pituitary gland will release these hormones into the blood, namely ADH and oxytocin. Oxytocin, the main targets, as I said, the breast during lactation and the uterus during labor. The ADH target is the blood vessels and the kidney tubules. And this is a more simplified diagram. This is the precursor, pre-pro-oxytocin and the pre-presophycin. This is the large molecule. It eventually will be broken down into neurophysins and hormones. The two hormones they're composed of 9 peptides, each oxytocin and arginine vasopressin, if you want to call it, or vasopressin or ADH. This one, the fragment of oxytocin is neurophysin 1, and the fragment of ADH or arginine vasopressin is neurophysin 2. And these are the ones which are released upon appropriate signaling. See, since the posterior pituitary hormones have similar structures, they have similar precursor, it is expected that they will exert more or less similar effect, but one of them is more effective in doing something compared to the other. If we take oxytocin and look what it does here, it's a primary action actually on the smooth muscles of the uterus during labor, something known as oxytocic activity. Its oxytocic activity is pretty high, where it also goes to the mammary gland and cause milk ejection. It has a high potency there, whereas its effect on the blood vessel is very little, and its effect in causing water reabsorption in the kidney is very little. If you take arginine vasopressin or ADH, you can see it may have some oxytocic activity, but it's very minor. It's not that potent. And again, it may have some effect on the mammary gland, but it's very little. But it has high or significant effect on the blood vessels. It causes vasoconstriction, and thus it increases the total peripheral resistance. And you can see that it has a potent effect as an antidiuretic hormone to increase water reabsorption in the kidney. If you take this guy, desmopressin, or DAVB known as, just remember the word desmopressin for the time being. This is a synthetic analog of ADH. It does not have oxytocic activity. It does not cause milk ejection. It has a very little effect on the blood vessel. However, it's a potent ADH-like hormone. I mean, it has a significant effect in increasing water reabsorption by the kidney. And that's why this guy sometimes is used to treat people who have abnormalities or unable to synthesize or secrete ADH. We'll talk about this in diabetes insipidus. Okay? Just keep it in mind. Now, these are the primary actions of ADH. It regulates osmolarity of the plasma or the blood by increasing the water permeability of the late distal tubules of the kidney, of the nephron, and the collecting ducts. Now, you will learn about this a little bit more when you take the renal system next semester. And the way by which it increases water permeability, it acts via V2 receptors, and they are J protein coupled receptors, and they work through the adenyl cyclic AMP mechanism, which will basically cause the synthesis of water channels known as aquaporin channels. Aquaporin channels in the principal cells of the distal tubule and the collecting duct. They make more channels. These channels will be inserted on the epithelial cell membrane, and thus water will get in. This is a complicated process. Just remember the action of ADH for right now. And it also causes constriction for the vascular smooth muscles. And I talked about this in my previous lectures. It acts on V1 receptor, and it activates phospholipase C and the activation of IB3 calcium second messenger mechanism. And as a result of increasing the constriction of the smooth muscles of the arteriole, basically it causes an increase in vascular resistance. And this is very important sometimes when we have hemorrhage. It contributes to the maintenance of blood pressure. Now this slide shows you, a schematic diagram of the one nephron. This is the glomerulus. This is the Bowman's capsule, proximal tubule, lobe of Henle, descending, ascending limb. And this is the distal tubule. This is the late distal tubule right here. That's where ADH works to increase water permeability. And this is the collecting duct, and it increases water permeability here. And the way it does it is as follows. Now that's the cellular mechanism of action of ADH. When we talked about the general mechanism of actions of hormones, this is a peptide hormone. It will interact with the J protein coupled receptor, the JS, and then it will activate adenyl cyclase. Adenyl cyclase will lead to the conversion of ATP to cyclic AMP, and this will activate protein kinase A, and then it causes phosphorylation and the synthesis of these aquaporins or water channels, and then they will be inserted within the membrane. And when we increase the number of water channels, water reabsorption is gonna be increased. This refers to the lumen here, the collecting duct lumen, or the distal tubule lumen. Just talking now about how ADH secretion is regulated. Sometimes we need to increase the secretion of ADH. There are certain things that stimulate ADH secretion and things which inhibits ADH secretion, like any other hormone. Now, the most important thing here which modifies the secretion of ADH is basically change in plasma osmolarity. A small increase up to 1% in plasma osmolarity, or osmolarity if you want to call it, of the ECF bathing the hypothalamic osmoreceptors. There are osmoreceptors. They are neurons which detect the osmolarity. They will evoke or increase the secretion of ADH. This pattern of regulation, we can call it osmoregulation, because the change occurs as a result of change in the plasma or ECF osmolarity. And if there is also a decrease at times, if we are overhydrated, the plasma osmolarity will decrease, and this will also decrease the secretion of ADH through these osmoreceptors. And that's basically how we maintain our homeostasis in terms of our osmolarity. And you know that the plasma osmolarity from your body fluids lecture is about 285, 290. That's a range. It could be up between 270 and 300. But that's about an average value. The plasma osmolarity, or the ECF osmolarity, is about 285 milliosmol per kilogram. And if you want to talk about in terms of osmolarity, it's gonna be 285 milliosmol per liter. And I'm sure that you know the details from your body fluid lectures in the general physiology course. The osmoreceptors are located in the hypothalamus, primarily in an area known as organum vasculosum of the lamina terminalis in the third ventricle. That's something you learn about it when you study your neuroscience course. Just keep it in mind for right now. Just remember them. And then these osmoreceptors are neurons. They respond to increase in plasma osmolarity or decrease in plasma osmolarity. In other words, their activity will increase when there's an increase in osmolarity, and their activity will decline when we have low osmolarity. And basically they detect the plasma sodium concentration in the fluid bathing these neurons in the hypothalamus, and they send synapses, okay, or they make synaptic connections with the supraoptic nucleus and paraventricular nucleus, and thus they stimulate the release of ADH from the neurosecretory cells in the hypothalamus and the posterior pituitary gland. And osmoreceptors also have another effect. They stimulate thirst. And you probably know by now from your general physiology that there are centers which regulates food intake, water intake. So the food, there are thirst centers, or there are certain neurons in the hypothalamus which actually induce thirst and and make us drink more water in a sense. So primary thing which causes an increase in ADH, basically an increase in plasma osmolarity. However, there is another important thing that we should know, that sometimes if we have volume depletion, and we can call this, if you want, volume regulation, if for some reason or the other, due to diarrhea, due to vomiting, due to bleeding or hemorrhage, as you may know, if we have hemorrhage, for example, and the blood level is reduced, or the amount of blood is reduced by more than 10%, 15 to 25%, this is a strong stimulus for ADH. And of course, as you may remember, the decline in the blood volume will cause a decline in the cardiac output, decline in the blood pressure. And you have atrial stretch receptors that detects the blood volume. We have the baroreceptors, which are located in the carotid, and the aortic receptors. The baroreceptors, they will initiate the baroreceptor reflex and this sort of thing. You have to go and revisit it. And apparently through these either stretch receptors in the atrium or the baroreceptors or efferents from the baroreceptor, somehow they will reach the hypothalamus and they will induce the release of ADH. And this is very important to maintain the blood volume in case of hemorrhage till we, resuscitate the patient and give him some fluid to maintain his fluid balance. Now, this slide basically shows you the effect of volume changes, of the ECF volume changes and changes in the osmolarity on ADH secretion. Here is the ADH level, and this here the plasma osmolarity. Let's just talk about the normal case where we have normal blood volume. For one reason or the other, if our plasma osmolarity is increased due to salt intake or whatever it is, the plasma osmolarity will increase, and this will evoke an increase in ADH secretion. If you, for example, if you drink lots of water, the plasma osmolarity will go down, and then you will decrease, the ADH secretion will decrease. So ADH is there basically to maintain the plasma osmolarity within this level, so it doesn't go too much beyond 290s, so that's basically what it is. Now look at this. If you have volume contraction, particularly if you have hemorrhage or bleeding, if you want to call it, here we have an isoosmotic dehydration. In other words, there is no change in the plasma osmolarity, but the drop or the decline in blood volume during hemorrhage will evoke secretion of ADH even if we don't have a change in osmolarity. So you can see here the osmolarity due to hemorrhage is not going to change because during bleeding we lose salt and water, but the blood volume is going to decline. So to maintain the blood volume, we have to reabsorb more and more water till we manage the patient. So ADH will be evoked, ADH release will be evoked in an effort basically to maintain as much water or to retain as much water as possible by the kidney. Now here, if you have volume expansion and a volume expansion, even if there is an increase in osmolarity, the response to ADH will be less. So here you have a case where you have volume expansion. In spite of the increase in the plasma osmolarity, still the level of ADH secretion is not as much as it is under normal conditions. Now, these are some of the factors which either increase or decrease arginine in vasopressin secretion or ADH. These are again increased serum or extracellular fluid osmolarity. They are the same. Decrease the blood volume or decrease ECF volume in general. Decrease in blood pressure, pain, nausea, vomiting, stress. Some drugs, you will learn about these drugs. Just keep them in mind for right now. Also, there are factors which inhibits ADH secretion. We just talked about them. Decrease ECF osmolarity, decrease serum osmolarity. They are synonyms. Increase ECF volume or volume expansion. And ethanol اللي بيشرب كحول بروح الحمام أزيد. It is possible atrial natriuretic peptide also cause inhibition when we have expansion of the ACF. And this is a hormonal mechanism. Atrial natriuretic peptide is released by the atrium whenever we have expansion and stretching of the atrial walls. Now, these are the primary disorders of ADH. When we have problem with either ADH action or ADH secretion, we will have something known as diabetes insipidus. بالعربي بسموه السكر الكاذب. هذا السكر ما بيكون عندنا we have normal glycemic control, we don't have diabetes, but still people they have symptoms of polyuria, polydipsia, they are thirsty, they drink a lot of water, they urinate a large amount of urine, etc. And that's what diabetes insipidus is all about. Sometimes it's caused by problems within the hypothalamus particularly, and that's why we refer to it as central diabetes insipidus. At times the ADH is there, the posterior pituitary is fine, the neurosecretory neurons are fine, but the problem there is like maybe mutation in the receptors, which are the receptors of the distal tubule and the collecting duct of the kidney. They don't respond to ADH. So there is like a receptor problem. It's not a hormonal issue, it's a receptor issue. And since the problem within the nephron itself, we refer to it as nephrogenic diabetes insipidus. These are the primary causes, or the main primary disorders of ADH secretion. At times we have tumors, for example, in the lung, which secretes large amount of ADH, and in this case we have, in a sense, hypersecretion of ADH. This is though a secondary disorder rather than a primary disorder, because the tumor releases ADH, and it's called as it is known as inappropriate ADH secretion, SIADH. and it's caused usually by, for example, like lung tumor. Here we have decline in osmolarity, expansion of ACF, dilution of proteins, among other things. Very rarely, sometimes in mothers with gestational diabetes, some mother may exhibit symptom of gestational diabetes insipidus. It is rare. It only occurs in pregnancy, and that's why we refer to it as gestational diabetes. Apparently, there is an enzyme in the placenta that is produced during pregnancy and destroy ADH, which is secreted by the mother posterior pituitary gland. Though this is not really, you don't observe it, it's very rare, just to remember it in case you run into a case. And these are the causes of central diabetes. It's basically characterized by deficiency of ADH in the plasma. Its synthesis and secretion, it's not there. Hypothalamic destruction by hypoxia, stroke, head trauma, infection, tumors like craniopharyngioma. I just mentioned this in my previous lectures. Here, as you can see, you have increased serum osmolarity because we lose a lot of water. We have dehydration, and that's important to remember. Polyuria, polydipsia. The urine becomes less yellowish أو بصير يعني مائل لون. إحنا عادة لونه yellow اليورين. بصير في عنا dry skin وبصير عنا change في السكين turgor as a result of dehydration. We may have constipation, we may have muscle weakness, and bedwetting during the night time because, especially in children. In nephrogenic, here, as I said, there is nothing with the pituitary gland, not wrong with the pituitary or the hypothalamus. However, the principal cells of the collecting duct or the epithelial cells known as principal cells, they are unresponsive to ADH due to defect in the V2 receptors or the GSA adenyl cyclase coupling mechanism. It might be caused in some individuals by genetic autosomal recessive aquaporine channel gene alteration. It's X-linked recessive V2 receptor gene alteration. ADH levels are elevated though. You can see this in this group of people. ADH is fine, and actually if you measure ADH level, it's going to be high in spite of dehydration and in spite of increase in plasma osmolarity, because the patient is using a large amount of urine. There is dehydration, there is increase in osmolarity, there is a decline in the ECF volume, and all of these tend to increase ADH. But still, the patient have polyuria, polydipsia, etc. And there are so many causes of this. Actually, I was reading. I don't want to overwhelm you. This is probably the most common, though. But we have sometimes some metabolic changes that may affect the concentrating ability of the kidney, like hypocalcemia, hypercalcemia, lithium. And apparently, these somehow diminish the effectiveness of action on the principal cells, but how they work is still not known. But apparently they may disturb the trafficking or the sequestering and the insertion of the water channels in the luminal membranes of the principal cells. Sometimes if you have chronic diseases of the kidney or renal infarction, and here because the thing is, the ADH is there, but the kidney tubules are not working. If you have kidney infarction or chronic kidney disease causing the damage of the kidney tubule. So in spite of ADH, the fact that ADH is there, the kidney is not responding because it's damaged. Sometimes I leave this, I hope I'm right, they give them thiazide diuretics because if you give them ADH, it's not going to help them. So you want to get rid of the excess water, and you may have to do something about this. You will learn this, and the pharmacology people will teach you more about this. Now in this slide here I am going to describe a test that you may want to use. if you suspect diabetes insipidus in a patient, and you want to determine is it central diabetes insipidus or nephrogenic diabetes insipidus. So let's say what you do here, you restrict the fluid overnight. The trouble is you have to worry about dehydration, especially if you have a child with a disorder. You have to keep, monitoring his weight and see how much fluid he is losing. And with restriction of fluid in someone who she or he has diabetes, it's going to be a problem. So you have to keep the patient under monitoring while you are doing this test. So let's say this is the normal person. You measure the plasma osmolarity. It's 297. It's fine. Urine osmolarity is normal. And you measure the ADH level, the plasma. It is there. I mean, we have ADH after fluid restriction in a normal person, because fluid restriction will cause an increase in osmolarity, and as a result of this, ADH will be increased and the urine osmolarity will be more or less maximum here. And now if you give the zumpresine, which is an ADH analog, the osmolarity is not going to change anyways, because he already has ADH. Now, if you take a patient with central diabetes insipidus, due to dehydration and the loss of water and the polyuria and the excretion of large amounts of diluted urine, you can see the plasma osmolarity is high. However, the urine osmolarity is low. Okay? And if you measure the amount of ADH, it's gonna be low. Even the patient is under fluid restriction. However, if you give this ADH analog, desmopresine, you can see after this overnight restriction, water restriction, and the administration of desmopresine, the plasma osmolarity increased, meaning that desmopresine works. Okay? So you can say that this patient is likely to have the central diabetes insipidus. Okay? Now, if the patient has nephrogenic diabetes, you measure the plasma osmolarity and you measure the urine osmolarity. Now you can see here, as a result again of dehydration and the excretion of large amount of urine, we have dehydration, the plasma osmolarity is high, whereas the urine osmolarity is low. But if you measure ADH in this patient, it's going to be high, because there is a change in the plasma osmolarity. ADH is secreted, but the nephron, or the collecting duct, and the distal tubule are not responding to ADH. There is a receptor problem here. In spite of the presence of ADH, ADH is not working. Still the patient, in spite of the fact that ADH is working, it's there, but it's not working. So you expect these things. Now, if you under fluid restriction here and you give this desmopresine, and again after giving this desmopresine, still the patient is excreting large amount of urine, and his urine osmolarity after the administration of desmopresine is low, meaning that in spite of giving exogenous ADH analog desmopresine, there is no response, and this will confirm basically that this patient has nephrogenic diabetes insipidus rather than central. Of course, you have to do other testing. If you suspect central diabetes insipidus, you have to do MRI of the brain, to figure out if there is a lesion in the hypothalamus, or you have to look at other things to confirm further the diagnosis. But that's something you have to know. Sometimes, if you have partial central diabetes, and there is a little bit of it, there is a little bit of ADH. The symptoms are not that severe, and the effect will be better when you give desmopressin, which is the ADH analog. Now the last thing I want to talk about, the syndrome of inappropriate ADH secretion, SIADH. Here we have excessive release of ADH due to a tumor, and there is excessive or increased water reabsorption. As a result, we have fluid expansion or ECF expansion, and we will have a decline in plasma osmolarity, and sometimes that's why we refer to it as hypoosmotic volume expansion. ECF osmolarity decrease. ECF and IECF volume increase because of the water retention, so there is also an increase in body fluid volume. The plasma protein concentration decrease because of the increase in the ECF volume. Increased water retention. This is very important to remember, leads to hyponatremia in spite of the normal sodium stored. In other words, the amount of sodium in the body is not changing. What causes hyponatremia here is basically the dilution of the ECF. And here we have inappropriate concentration of urine. Usually the urine concentration is so high, and it's much higher than the plasma osmolarity. Some of the causes of this is ectopic production of ADH by tumors like small cell lung carcinoma, drugs, some of the drugs like carbamazepine, which is one of these anxiolytic drugs. You will learn about this. You don't have to worry about this at this point until the time you learn about it. The principles of management of different types of disorders of ADH. In SIADH or syndrome of inappropriate ADH secretion, you try fluid restriction, especially if the hyponatremia is mild. However, if you have severe illness and there is hyponatremia is significant, you may want to give hypertonic saline to try to boost the levels of sodium in the plasma. And you want to give a V2 receptor antagonists. There are some of them here. You will learn more about them in pharmacology. I'm sure they will teach you this. Sometimes they give an antibiotic, which is amazing, democyclocycline. It's a tetracycline. Demeclocyline. It's a tetracycline, but somehow it decreased the responsiveness of the collecting tubule cells to ADH. In case of central diabetes, you give desmopressin, DDAVP. It's an ADH agonist or analog, so you will help them out. In nephrogenic diabetes insipidus, this will not work, so sometimes you want to give them a diuretic, like thiazide diuretic. The pharmacology people will teach you better, and they know better than I do about all of this business. So now let's go to oxytocin. We touched on this a little bit when we talked about the prolactin, if you remember, if you have looked at this lecture. As you may remember, these are the hypothalamic nuclei, the paraventricular and the supraoptic. The source of the main source of oxytocin is the paraventricular nucleus. The supraoptic nucleus in neuron release a little bit. Again, it's synthesized here as a pro-oxyphysin, and it's broken down to oxytocin and its physin fragment. Physin 1 fragment. Now you can see the main target is the mammary gland here, and what it does during lactation, when you have suckling in the nipple, the signal will be transmitted to the nervous system, to the hypothalamus, stimulate the release of oxytocin, and it goes to the mammary gland and causes contraction of the smooth muscles or contraction of the myoepithelial cells and the squeezing of the milk into the duct system of the breast. And another primary function of oxytocin is during labor. We don't know how labor starts, but at the end of term, after like 37, 38, 39 weeks, usually dilation of the cervix and the plugging of the head or descent of the head into the cervix. It causes stretch of the cervix of the uterus, and these receptors or the stretch receptors will send a signal to the nervous system via complicated pathways that you may learn about them when you take the CNS, how we transmit mechanoceptive sensation in our body. It goes to the hypothalamus and stimulates the secretion of oxytocin. And here what oxytocin does, it causes contraction of the smooth muscles of the uterus and, aid in labor. And, also during labor there might be the release of another hormone, prostaglandins, and these also considered to play a role in labor. And a lot of time actually some women may receive oxytocin injection to stop the bleeding after delivery, postpartum management so to speak. يعني إذا واحدة صار عندها شوية نزف زيادة ممكن نعطيها أوكسيتوسين، لأنه الأوكسيتوسين بيساعد على to stop the bleeding maybe through its vasoactive action or so here are some of the effects. The milk ejection or a milk let down reflex I just talked about. Sucking the nipple will send a signal to the hypothalamus. The hypothalamus will release the oxytocin. It goes to the mammary gland and cause the contraction of the myoepithelial cells. And of course, the milk will be released and it will be ejected eventually. It goes through the lactating breast into the large duct system and thence to the nipples, and that's known as the milk let down or milk ejection reflex. Sometimes actually people may start ejection of the milk without even suckling of the breast, and they may have spontaneous discharge of milk. Sometimes if a mother thinks of her baby while she is working or something, so this is anticipatory, or when she gets home and see her baby or something if she is working. Sometimes the sight or the sound of an infant when he is with his mother, when he cries or something, it may affect the hypothalamic neurons of the mother and cause the ejection of milk. Now the role of oxytocin in the male is not clear, but there are evidence which suggests that oxytocin increases at the time of ejaculation of the semen. So it's possible that oxytocin go to the vas deferens where and it causes the contraction of the smooth muscles of the vas deferens to get the sperm into the urethra or toward the urethra. Though that's not a confirmed function. And during labor, I just mentioned this, oxytocin causes contraction of the smooth muscles of the uterus involved in labor and parturition. And the sensitivity of the uterine musculature to oxytocin is enhanced by estrogen and inhibited by progesterone. That's why we don't want oxytocin to cause any contraction during the pregnancy. And once we have high levels of progesterone, usually the effect of oxytocin is not that significant. However, at term, the level of progesterone drops, and that's how oxytocin starts to work. Now, which is at the at the end of the pregnancy. Actually, the uterine cells, smooth muscle cells, become sensitive to oxytocin, and that's probably what contributes to the labor. Also, oxytocin is then increased during the labor, like as I mentioned, due to dilation of the cervix. And oxytocin can be used to induce labor, okay, and reduce postpartum bleeding. يعني مرات في ستات بتأخر الطلق عندها وبدنا نعطيها إشي نبديه بعطوها oxytocin أو prostaglandins. Oh, there is a possibility that oxytocin may act non-pregnant women in the uterus to cause some contractions of the smooth muscles of the uterus to help the sperm travel up to the fallopian tube so that fertilization can occur within the fallopian tube. Other function, I just mentioned this. So it's repeated. As I said, it could be involved in the propulsion of sperm toward the urethra during ejaculation. It's believed to increase it during orgasm, which, after sexual intercourse. It could be increased by stress, and it's again inhibited by alcohol, like ADH. And actually, there are studies which believes that this hormone is good for our positive social interaction. It has been involved. It has some emotional-like effects, like, when you feel good about seeing somebody or when you love somebody. And just to mention something that oxytocin disturbances have been associated with autism or autism spectrum disorder. Autism, what's autism in Arabic? It's, I don't know, I forgot what's autism in Arabic. توحد, مرض التوحد. Now, this is a case for you to read. Okay, it summarizes things about central diabetes insipidus. And, I think when you read through this, you can figure out if you have learned how ADHs work. And I think there is another case that follows. And please read these cases and go over them, because you can put things together. You see how much you know, how much you don't know. And this is an approach that you have to take when you study these cases, because that's what's important. It's not just to memorize the function of that hormone, how it does, how it works. You have to apply this knowledge in a clinical context. And that's why basically I posted these cases in my lecture, so that you start to think how to put things together, how to use your basic science knowledge in a clinical context. And because that's the end of the day what matters, really. You can list 10 factors which may affect ADH secretion or inhibition, and the same token by oxytocin. But you have really to know the importance of these things. When you go to the hospital, when you go to your residency, you have to think about it. And this will help you actually if you put what you have learned in basic sciences like physiology and other areas in a clinical context. And that's because you are going to be a doctor, nobody's going to ask you, what's ADH at that time, because they assume you know it. But you have to utilize this knowledge to be able to understand what goes on. If you have a clinical case, you want to know what it is, how to deal with it, what's the cause of it, and so on and so forth. Thank you very much.